is share my screen with you. Can you see the student questions, coronavirus?
Yes.
Yes.
Okay. All right. Make it bigger. Oh, so what's the point? Why did I do this? Well, it's an experiment, and I'm trying to see if this is a study tool for you, if it helps you learn by thinking about what you don't know and asking questions about it and then discussing it in class. If it works, it's a good expenditure of time. We'll find out. Okay, the first several questions I want to deal with are of a sort of a basic and general nature, starting with several people ask about RNA and specifically about the ribosome. So I refer you to this slide, which shows the three types of RNA that we talk about mostly, including this picture showing each of the three types, the mRNA, the rRNA, and the tRNA. So in particular, what's the difference between the ribosome and the rest of it? Well, the ribosome is like a little machine. It has two parts, the large subunit and the small one. And they combine to make proteins based on the message in the mRNA and the tRNA, which brings the amino acids. So if we go down to the next slide, it shows these components in action. Up in the upper left-hand corner here, you see the tRNA, the large and small subunits, and the messenger RNA. If you go to the right, you can see them all combined. The two subunits of the ribosomal RNA contact the mRNA. The mRNA is sandwiched in between them, and then it moves from codon to codon down the chain. And as it goes, for every codon, it calls for another tRNA, depending on what that codon is. And that determines what the next amino acid in the chain is. You can see that if you go down a notch, you can see two different tRNA molecules there, each carrying a different amino acid. They're stitched together by peptide bonds. And in the end, the peptide goes off, and the components of the machinery can be recycled to start all over again. So does this help? Does this help make the distinction among the three different types of RNA that are involved in protein synthesis, how they work, how they collaborate, come together to make a protein?
Yes.
Okay, clear? Then we'll go to the next question. This question was about the 6 steps of translation. I think what the person was really asking is the 6 steps of DNA replication. And to address that, first of all, let me refer you to the video that we saw. You can find the URL for that in the slide. But what I have here is a screenshot from that video. which summarizes the steps in DNA replication and the factors that are involved. So the first enzyme is helicase, which unwinds the two strands. And then you have the SSBs, which keep the two strands apart, keep them from collapsing back together. And then there's an enzyme called primase, which puts an RNA primer, and it's important to emphasize it's an RNA primer, not DNA. But the little red mark here in the drawing is a primer. So there's one on one strand, there's another on the other strand. The primer is necessary because the big player here, that actually does the replication, DNA polymerase 3, cannot start on its own. It has to have a primer in order to get going. So these four components are necessary for synthesis on either of the strands. Two of them are used only for the lagging strand, in addition to the DNA polymerase, of course. But they are RNase H, which removes the primer, and DNA ligase, which joins Okazaki fragments. So what you see is the so-called replication fork. And remember that synthesis is always from the 3' to the 5' end. And so on the so-called leading strand, synthesis is continuous. It starts at the 3' end with the primer there, and it continues, never stops, goes all the way to the end of the molecule. Never has a problem. Now, the lagging strand does have a problem. And why is that? It's because as the helicase keeps unwinding the two strands, and it's moving from left to right, remember, DNA polymerase can only copy from 3' to 5'. And so in order to do that on the lagging strand, it has to go backward. And so that process is discontinuous. As a new section of DNA is unwound, a new primer goes down, DNA polymerase makes a short stretch of DNA, copies DNA. The primer gets removed by RNA H, and the two strands that have been just made are joined by DNA ligase. So all this extra work has to be done because of the requirement that DNA polymerase has to go from 3' to 5'. And that's shown graphically a little more neatly in the next diagram, where you have the leading strand and lagging strand. The DNA polymerase, the big green ball, is just going continuously on the leading strand and discontinuously on the lagging strand. And you have the addition of the primers keep having to be made, and DNA ligase has to put the Okazaki fragments together. So for those of you who are wondering about DNA replication, is this clear? And I should say it would be good for you to go back and review the video. Video goes through all of these steps in great detail. No questions?
Excuse me, Professor, I have a question about replication. So the helix, it separates the hydrogen bonds, right? So
Yeah.
why it's the first step? I think the first step is when it start like the helix become two strained before it's a break. So helix, it's break the hydrogen bonds between the nucleotide. But I think it was the topspimers that help the helix become too strained. It's the first one before we start to break the hydrogen bonds.
Well,
So
there's
so
no
why it's
there's no space. If you go look at this diagram to the far right, you'll see the hydrogen bonds are all there, and there's no space there for DNA polymerase to get in and do anything. And so those bonds have to be broken and make a replication fork.
but
first.
before the broken, it has to be two strand, or they will broke first.
I'm not sure I understand how you're visualizing this. DNA polymerase is trying to copy both strands.
Yes,
but it doesn't have room to get in to do the copying until the strands are separated, and separation calls for breaking of the hydrogen bonds.
yeah, but what I'm saying, the first step is they are helix, right? So just it's the helix became like straight line.
So are you asking about topoisomerase and how it
Yes.
untwists?
Yeah.
Okay.
Yeah.
So I think
it has
that's
to be
where
the first.
I think that's where she's coming from. The actual DNA replication starts here with the helicase, but so this is what you will commonly see referred to as the 6 steps of replication. However, you are absolutely right. Topoisomerase is critical in uncoiling the DNA, but just
Yes.
uncoiling the DNA has no real. It's something that happens anyway. It's not something that is specifically part of replication. So when you're talking about because topoisomerase can unwind DNA for many things as it's needed. So that's not necessarily a true absolute replication step, but this is. So just in this case, assume that helicase is step 1. I do see what you're saying, and congratulations. It's good of you to have studied and you know about topoisomerase.
Yeah. Okay, thank you. So the first step, it has to be helicase.
For replication.
Yeah. Okay, thank you.
Okay, anything else on DNA replication? Okay. That's a good question. Probably come up on an exam, so make sure you understand all the steps. Now, there were several questions about mutations, types of mutations, and how to recognize them. So that's what I want to address next. I refer you to this slide from the lecture, types of mutation. And what I'd like to do is illustrate each type with this sequence. This is actually a sequence that you had on the last quiz. So there's a wild type sequence here. It's just random A's and T's and G's and C's. And the next strand illustrates a deletion. So how do you recognize that this is a deletion? Well, if you go along and you compare the mutant strand with the wild type strand, it's identical. All the A and A, C and C, A and A, T and T. You go on until you get down to GGA, and suddenly in the mutant strain, it's not there. But then if you go on, you come back to C and C, A and A, T and T, and it's identical. So the only difference between the mutant and wild type is that this GGA is deleted. And so it's called a deletion mutation. What about a duplication? Well, if we do the same thing, comparing wild type with mutant, the sequence is the same as you go along. The AACCTT is the same. GGA is the same, but then instead of going to CAT, the next codon, you get GGA again.
It's duplicated.
So the GGA sequence is duplicated. It's copied and repeated, and that's called a duplication. And then the sequence goes back to normal, CAT, CGT, and so on to the end. So that's illustrative of a duplication. And I'm illustrating these with 3 letters at a time. They don't necessarily have to be 3 letters. It can be anything from 1 or 2 or 3 letters up to kilobases. Okay, what is an inversion? An inversion is where a piece of DNA flips over. So again, compare wild type with mutant, and it's the same as you go down the strand until you come to the GGA. Instead of GGA, it's inverted, it's AGG. And then after that, it's the same. So that is how you would recognize a mutation as an inversion if the sequence suddenly starts reading backward. And finally, an insertion is just that. Again, we compare wild type with mutant, and we get down to our GGA codon. And then instead of the next codon being CAT, it's AGC, TCG. But then it comes back to the wild type sequence. And so these 6 letters are an insertion. So does that help to clarify how you read a sequence and how you can tell what type of mutation it is? This is important. We'll probably deal with this again on tests.
Excuse me, teacher, this is the wild type. It's not a normal type. How we compare these kind of mutation with this wild type? It's a normal sequence?
Wild type?
Yeah.
Yeah, wild type is by definition. You choose a sequence that you think is normal, and that's your standard. You compare every mutation with that standard sequence.
Okay. Oh, thank you.
But there's nothing sacred about the wild type sequence. It's whatever you, the investigator, choose it to be. Okay. There were several questions about macromolecules. The essence of it is, what are the 4 components of macromolecules? So what are they?
And they are 4. They are protein, carbohydrates, lipid, and nucleic acid.
Okay, so those are the 4 macromolecules. But this question, at least as I understand the question, is what are the components of these macromolecules? What are the atoms the macromolecules are made of?
What are the 4 components of? Teacher, they are different from the when it's protein. They are made from amino acids, from monomers like amino acids. And when they are lipid, it's different from each one.
Yeah, what are the elements
The elements
that go into macromolecules?
they are monomers.
Yeah, but what are the monomers made of?
Yeah, it's for lipid. For like carbohydrate, it's monosaccharides.
Yeah, but what are monosaccharides made of?
Monosaccharides made up carbon, hydrogen, and oxygen.
Right. So that's one of them. And then if you consider nucleic acids, what's an additional element in nucleic acids or protein?
Nucleotides. They're
And what are they made of?
nucleotides. The nucleotides, they are like yeah, they have three components: nitrogen base, phosphate bond,
Okay, you've identified.
carbon.
So remember the acronym CHON, CHON, carbon, hydrogen, oxygen, and nitrogen. Those are the components of macromolecules.
Dr. Yoder,
Yeah,
would you also include phosphorus and sulfur? Because sulfur is found in a couple of the aminos, and phosphates are found in sugar carbon backbones.
yeah, they are certainly components and major components, but the four that are usually indicated in macromolecules are the acronym CHON.
Yes, but vast majority, and I absolutely concur. I was just making sure that adding in those other two was also, you would consider that valid.
Yeah, that is valid. All right, so let's move. I've divided the rest of the questions into sections: membranes, coronavirus, and cystic fibrosis. And first, I'd like to consider membranes. There were several questions about what amphipathic means. The concept of it is very simple. It's a linear molecule, which is polar on one end and nonpolar on the other. So that is an amphipathic molecule. And to illustrate that, I gave you three examples, which I've got listed here. One is in the phospholipid bilayer. The phospholipids are amphipathic. If you look in this little diagram, the long gray chain is a nonpolar tail, and the pink and blue other end is the polar head. And why are those necessary? Well, they're necessary to make a membrane because the nonpolar tails attract each other, and the polar heads are exposed to the external environment, which is always water, always aqueous. So this is an example of a cellular structure that requires an amphipathic molecule. The other example, we were talking about cystic fibrosis. Again, amphipathic came up. Why? Well, it has to do with digestion. And we considered this bile acid, glycocholic acid, which is amphipathic. It's very nonpolar on one end, it's a steroid. The other end, it's polar because it's an amino acid. So it's polar on one end and nonpolar on the other. And what is the function of being that way? It's to allow digestion of fats. All the components of the food you eat are compatible with water. They're water soluble, but fats are not. But yet they're a major component of our diet. So how do we extract energy from fats? Well, first we have to make them compatible with water. And that's what bile acid does. It first emulsifies the fats and then solubilizes the emulsions so that enzymes, which require water, enzymes can act on them and extract energy from the fats. So that's the second example of an amphipathic molecule and what it does, why it's necessary. The third is soap. When you wash your hands, amphipathic molecules get your hands clean. Again, a soap molecule is a long chain, a hydrocarbon chain, with a hydrophilic head. And when you wash your hands, you have oily substances on your hands, micelles form. And all the hydrophobic tails go into the oil or whatever substance you have on your hands, and the hydrophilic heads are sticking out into the water. And that allows the structure, the micelle, to lift the dirt off your skin into the water where it gets washed away. So next time you wash your hands, think about what's actually happening. It's an amphipathic molecule. Okay, any questions about that? No? Okay, several other questions were about what can be transported across a membrane? Does active transport require a membrane? And what's the role of cholesterol in a membrane? So I'll try to deal with all of those with this one slide. Again, taken from a lecture. What are the factors that affect membrane transport? Well, they're listed here: temperature, fatty acid saturation, and cholesterol. And how do they affect it? Well, as temperature goes up and down, fluidity increases and decreases. As saturation of the fatty acid chains increases and decreases, fluidity changes along with it. So the more unsaturation there is, in other words, the more double bonds there are in the hydrocarbon chain, the more fluid the membrane is. And finally, there's cholesterol, which acts like a buffer. At low temperature, it increases fluidity, and at high temperature, it decreases it, which allows an organism to be more able to adapt to a changing environment. So at high temperature, cholesterol raises the melting point so that the membrane can hold together instead of falling apart. If the temperature gets too high, it falls apart and the cell dies. And at low temperature, cholesterol in the membrane, wedged in among these hydrocarbon tails, makes spaces, which allows the membrane to continue to be fluid, continue to be flexible at a lower temperature than it normally would be. The other part of this, does active transport require a membrane? I would say yes. I can't think of active transport across anything other than a membrane. So what can cross through the membrane and what causes it to cross? Well, there's a lot of information about this point. I chose this one slide to try to capture the essence of it. There are three possible types of transport across a membrane. There's diffusion, which is entirely passive. And you can see that the red balls are simply diffusing, and this is usually quite a slow process. There's passive transport, which again is diffusion, but it's through a channel in the membrane. And then there's active transport, which is distinguished by the fact that it goes against a concentration gradient and it requires ATP. So you can see the blue balls, they're at a low concentration inside, at a high concentration outside, but yet they're moving from inside to outside against the concentration gradient. And they're using energy to do that. The other two processes do not require expenditure of energy. So any question about that? I quoted something here. Gases such as oxygen and carbon dioxide can pass freely through the membrane. Water can pass, but very slowly because the inside of the membrane is polar. But it's assisted by the process of osmosis. And as you remember, osmosis is the movement of water through a semi-permeable membrane. Okay, let's move on to another aspect of membranes. And that, Scott, this is your area, explaining tonicities. So could you give us your version of how to distinguish tonicities?
Yeah, okay, you're hearing me. So when you think about these problems, you need to understand what they're asking about. When they ask you about osmosis, they only want to know about water and what water is doing. When they ask you about tonicity, they only want to know about solute and what solute is doing. So you need to keep those two things apart and separate from one another. The other thing about tonicity is you have to understand tonicity only applies when you are comparing two things. For example, if you have a glass of salt water and you want to know if it's hypotonic, you don't know because you have nothing to compare it to. But if you have a glass of salt water and a glass of fresh water, now you can compare tonicities. So tonicity by itself on a single thing doesn't really apply. It's just, for instance, in my example, it's just a glass of salt water. You can't tell tonicity until you're comparing it to something else. So what is tonicity? Well, if you look at the word hypo, it means below. And tonicity, I said, refers to solute. So you can kind of look at the word as being low solute. So which of these things has the lowest percentage of solute in them? Hyper means excited, above, over. And so you put hyper with tonic or tonicity, and you're asking which of these things has the high solute. Iso means equal. And you'll probably not see isotonic questions because nothing really happens, but you definitely need to know the difference between hypo and hypertonic. So in workshop, I can go over these in much greater detail, but you really need to understand when they say tonic, they're referring to solutes. So you have to look at solutes. Do not look at the water, look at the solutes. If you lay this out, I actually like to make a little diagram, if you will, and I will label each thing. For example, if we're talking about a cell in a beaker, the cell will have a certain amount of solute in it, and I will list that. And then I will have, the beaker will have a certain amount of solute. Now I will list that. And then I figure out what percent of the solution is the solvent, which is, it's easy because if you just have a solute, say your solute is 10%, then the rest of the solution is considered to be 90% solvent. It has to total up to 100. So if you could do a little bit of simple math and you lay these things out, how much solute, how much solvent you have in each of the compartments that you're looking at, then you will be able to figure out these questions a lot easier simply by looking and knowing that things go from high to low because these are all passive. These are all passive processes. So if you have, two beakers and a tube that connect them and a semi-permeable membrane, you just have to figure out, the problem will tell you what kinds of things can move through the membranes. And then you simply look at which things have the highest percentages. And if it can move through the membrane, it will move through the membrane from whatever's high to wherever's low. again, this is like super fast because you have a lot to go over, but that's the basics of hypertonicity and osmosis. It's not super hard. You just need to see it laid out once or twice and then play with it a little bit on your own, and you should get this pretty simply. Um, is that kind of what you're looking for, Dr. Yoder?
Yeah, it's a good explanation. Are there any questions for
Professor, I
Scott?
have an example for this. If we put a little salt in our hand and we keep it, it's maybe after a few minutes it become moist and water. Can we say it's an hypertonic in this time?
You would have to be comparing it to something else.
Okay.
So tonicity has to be a comparison between two things.
Okay.
That's why when you look at these problems, you'll always have like two beakers that are connected, or you'll have a cell that's in a beaker of solution, because the cell is one thing that you're comparing to the beaker contents, which is another thing. Or sometimes I've seen the plants are in the ground and the ground is wet, and so would the plants be considered hyper or hypotonic, because you are comparing the solute concentration inside a plant versus the solute concentration in the soil. So you always will be comparing two things when you're looking at tonicity problems.
Yeah, it's a very good point you're making, Scott. The fact that it has to be a comparison. Anything else on tonicities? Okay, there was a question, go down to the next illustration. There was a question about does an electrical gradient form across the cell membrane? And the questioner said, is it because of the differing concentrations of ions on either side? And I think the answer to that is yes. So if you look at the illustration, you can see that the concentration of sodium is much higher on the left side than on the right. And because sodium is charged, has a plus charge, and it's higher on the left and the right, then the left side of the membrane will be negative relative to the right side. So to look at it that way, the electrical gradient is a function of the chemical gradient. Scott, do you see it that way?
I do, yes.
Okay, any question about an electrochemical gradient and how it forms, why it forms?
So if you do online reading, you can find yourself really quickly taking a deep dive, and they start talking about different ions and membrane potentials. For Bio 130, you really just need to understand that yes, chemical gradients form because of this chemiosmosis, these ion concentrations on different sides of a membrane. And as ions move, the membrane potential can change. And that's very important for things like nerve cell firings. But for this class, you don't necessarily have to understand all of the nuances. Just what's on this slide is about as deep as you need to go. So my caution is, when you're trying to read, if you're looking online, you will probably quickly find yourself in information that is way more than you need for answering any questions in Bio 130.
Yeah, in other words, keep it simple. Just understand the concept. Okay, there were several questions about different types of endocytosis, pinocytosis, receptor-mediated, and phagocytosis, and what determines which one it is. Well, receptor-mediated endocytosis, just like it says, it requires a receptor. So a good example for you to keep in mind is coronavirus, which the virus has this spike protein on its surface. The cell has a receptor, ACE2, and when those two get together, it's a signal for the membrane to invaginate and take up the virus into a vesicle. So the signal there is the receptor ligand binding. In the case of pinocytosis, the signal is what's in the liquid. So it could be proteins, it could be any, any number of molecules, but what's in the liquid signals to the membrane to invaginate, pinch off, and make a vesicle, take up a sample of the liquid. And in the case of phagocytosis, you have a large object like a bacterial cell, which again signals to the membrane to invaginate, pinch off, and engulf the bacteria in a vesicle. Okay, I'm going to skip the rest of this. I'm going to come back to it if we have time. I'm concerned about running out of time because I want to talk about the main questions about cystic fibrosis and coronavirus. So what causes cystic fibrosis? You should know this. This is one of the questions that was asked a number of times, but we've talked about the cause. What type of disease is cystic fibrosis?
Inherited. It's a kind of inherited disease or genetic disease.
It's a genetic disease.
Yeah.
Strictly genetic. That's all it is. It's the only cause of cystic fibrosis. It's not contagious. Some people ask, is cystic fibrosis heritable? And the fact that it's a genetic disease means that yes, it is heritable. This diagram illustrates how it is inherited. So if you have two parents and one carries a mutation for cystic fibrosis on one chromosome, but not the other. The other parent is the same, it has one affected chromosome and not the other. Then during meiosis, you get this random assortment whereby 25% of the children, or a 25% chance, will be unaffected. They'll have wild type genes and therefore have no cystic fibrosis. 50% of the children will have one copy, good, and one copy mutated. So they will be carriers, but they'll have no symptoms. And then 1/4 of the progeny, 25%, got two copies of the mutated gene. They're the ones that show symptoms. So that's how cystic fibrosis is inherited.
Excuse me, teacher. This is the X-linked disease. It's related to X chromosome because of this.
No, it has nothing to do with sex.
X-linked, X chromosome.
Yeah, it's not on the X chromosome. This is on chromosome 7, if you recall. And the next topic, the next lecture will be about cell division and about sex-linked genes. Okay, there were some questions about what is the path of the CFTR protein in the cell. And for that, I point you to this diagram, which starts with the nucleus and ends with the plasma membrane on the right. So the CFTR protein is encoded in the chromosome in DNA, which is transcribed and translated into protein, if all goes well. And that protein then goes through a very elaborate processing, which involves adding sugars, both core sugars and complex sugars, in the Golgi apparatus. And then there's transfer through a number of vesicles until finally the protein is deposited in the plasma membrane. And what's important about this for you is that for your project, you will have a mutant. And that mutant is going to be defective at some point in this process. Starting either at the beginning, you may get no protein at all, or you may get a protein that's defective and it doesn't work well when it gets to the membrane. But there'll always be a defect in the mutant that you're studying. So for purposes of your thinking about this, refer to this, this picture, and you can watch a video if you go to this website. Okay, there were a number of questions about zymogens. What is a zymogen? Anyone?
It's an inactive enzyme.
Yes, it's an inactive form of an enzyme. Does cystic fibrosis affect the production of zymogens? And the answer is yes, it does. It causes zymogens to activate prematurely before they leave the pancreas. And so if we ask, why are there zymogens? You remember it's because it's the proteases that are in the form, that are produced in the form of zymogens. And that's so because proteases have protein substrates in the pancreas, and the pancreas would digest itself if it made active proteases. So the zymogens end up going into the small intestine, into the duodenum, and it's there that they're activated into active proteases and can participate in digestion. So the cystic fibrosis causes pancreatitis, which is an inflammation. And that's what activates the zymogens prematurely, and that can have a very bad effect on the pancreas as an organ. Well, can pancreatitis lead to diabetes? There have been studies on this. Overall, even though the evidence is not strong, it does seem as though insulin production is affected by, certainly by cystic fibrosis and by inflammation of the pancreas. There were a couple of questions about why pancreatic cancer is so deadly. And the reason is that unlike most cancers, it has no symptoms until after it's metastasized to other organs and it's too late. So people who contract pancreatic cancer don't know they have it until it's too late to do anything about it. In general, there are exceptions to that. There were a couple of questions about can a person live without a pancreas? And the answer is yes, but the only way they can do that is if they supplement the pancreas functions in their diet. So in other words, take dietary supplements that contain the hormones and the digestive enzymes that the pancreas would normally produce. And then there were quite a few questions about the treatment of cystic fibrosis by gene editing. Could you edit the DNA of a fetus and make that fetus, switch it from a cystic fibrosis patient to a normal person? The answer is, in principle, it could be done. Is it considered ethical? Not by most people. But certainly the CRISPR technology is applicable here. The problem, you can use CRISPR to edit DNA in test tubes, in cells in test tubes. Then the question is, how do you extend that to the whole organism? So this is the whole organism after development. It's not in the fetus. And so it all comes down to delivery methods. How do you deliver the genetically engineered wild type into a patient? And there are lots of approaches to doing that. For example, putting the cells in nanodrops of oil and having them fuse with the patient's cells. The use of adenovirus, which we'll talk about in a little bit with respect to coronavirus. So different types of editing, I should say, editing can be done, and there are different types of ways of imagining how to extend that to cure a disease. To my knowledge, there's been no examples of this with cystic fibrosis where a patient has actually been cured by gene editing. Some people asked about what are the symptoms. We talked about in detail symptoms of cystic fibrosis in the digestive system. But what other kinds of symptoms are there? Well, the lung is the other major organ that is affected. And so cystic fibrosis causes severe respiratory problems breathing, and it causes male infertility. Why do you think it causes male infertility?
This is Zach again. Isn't it that it causes malformation of the vas deferens?
Not necessarily malformation. You're at the right point in the body, but what does it do?
Or not, it just doesn't form. That's what I meant, sorry.
No, it forms. The vas deferens is there, but what's wrong with it in a CF patient? So what happens on the cell surface if you don't get chloride ion transport? With the mucus.
It's lost. It's been locked, and there's a lot of mucus, right?
Yeah, a lot of not only mucus, very thick mucus. So it blocks the vas deferens, and there's no transfer of sperm. So that causes male infertility. Because the disease causes an ion imbalance, osteoporosis is often a result. And a typical symptom is excessively salty sweat, again, because of an ion imbalance. And it all starts with the inability to transport chloride ions. There was a question about whether the effect of a mutation can be seen immediately. I'm not quite sure how to interpret that. It depends. So for example, if you were looking at antibiotic resistance in bacteria, you could see that effect immediately. As soon as the mutation occurs, the mutants in the presence of the antibiotic would immediately proliferate. In the case of, say, cystic fibrosis, you need the body to develop at least to a certain stage before you can see the effect of the mutation. And then we're getting short on time, so I want to go on. We can come back to some of these questions later, but I want to make sure we touch base on the most important ones about coronavirus. There was probably more interest in coronavirus than any other topic in this section. And one question it's a very simple question, but it's really important. What's the difference between SARS-CoV-2 and COVID-19? Well, SARS-CoV-2 is the name of the virus. There are a lot of SARS-type viruses. They're severe respiratory viruses, coronavirus being the most severe. COVID-19 is the name of the disease. So a person that has contracted this virus now has a disease called COVID-19. Several questions about the origin of SARS-CoV-2. Where did it come from? Well, it's my understanding that although a lot of studies have been done and continue to go on, it's not known conclusively. But the best evidence suggests that this virus originated in China, originated in humans in China, and it probably came from an animal. Some evidence that bats do carry SARS-CoV-2. Whether bats transmitted it to humans has not been shown conclusively as far as I know. Then there were a lot of questions about the vaccines. The 3 vaccines that are available in the US at this time. So let's just go through this list. What is the effect of the vaccines that we currently have against the new variants? In general, the new variants are all subject to all 3 of the vaccines. There's no variant that escapes yet, which is the next question. When will a new variant arise that will be immune to the currently available vaccines? Well, it's only a matter of time. The more times this virus copies itself, the more chances there will be for copying errors, mutations, and the more chances there will be for a new variant that the current vaccines will not be effective against. So that's why there's such pressure to get as many people vaccinated as possible, as quickly as possible, to cut down on the number of copies of this virus. For every new patient, it's a whole bunch of new copies, a whole bunch of new chances for variants. So where are the COVID strains coming from? They're coming straight from the patients that have the virus. As the patient replicates the virus, more and more variants arise. And there was a question about why is the Johnson & Johnson vaccine approved if it's only 66% effective, which is often reported in the media, whereas the other vaccines are 95%. Well, the numbers that you read in the media are usually not comparable because of all the variation that occurs in the testing, in the clinical trials that have been done. So it's 66% is not an accurate number. It's actually higher than that. May not be 95%, but it depends on the cohort that you're testing it against. So if you're testing it against people with serious symptoms, they're all equal. If you're testing it against people with less severe symptoms, one might be more effective than the other. So at this point, there's no reason to choose one over another. The recommendation is whichever one is available to you is the one you should take. Long-term side effects and will boosters be needed? Don't know. And the reason for that is this is a new disease in humans. It's only been with us a year, and we don't have not had enough time to determine the long-term effects or whether booster vaccines will be needed to maintain immunity to the virus. Can people die from getting the vaccine. I've never heard of such a thing. I don't know if any of you have, but as far as I know, there have been no deaths attributed to being vaccinated. Several people ask, why do you inject into the muscle? Well, ask yourself, how many options are there? If it's not the muscle, what are you going to inject in? So I think you could put the vaccine into other tissues, but none is as available as a muscle. And a muscle is very densely packed cells for the vaccine to enter into. And it's convenient. You have a big muscle in your shoulder. It's easy to inject there. Some people asked about why there can be a reaction, in some cases a severe reaction, to the second shot. I actually experienced that myself. And it's my understanding that with the Moderna vaccine, which is the one I had, that they've included an adjuvant in the vaccine, which stimulates the immune system. It's not an antigen, but it just primes the immune system to react to the antigen. And the reaction of your body is actually to this adjuvant and not to the vaccine itself. Our vaccine's mutagenic, not to my knowledge. Can people with evil intentions alter other people's DNA by injecting them with a vaccine? I don't see how that could possibly happen, and I don't think it is happening, or that anyone, as far as I know, no one is attempting to do that. And finally, do young people need vaccine? The answer is yes. Even though the priorities for most jurisdictions are that older people get vaccine first, young people can get sick and die, not at the same frequency, but certainly it's not worth the risk. And then there were several questions about how vaccines work. So let's just go over that again by referring to the slides that I showed. How do mRNA vaccines work? And do vaccines contain antibiotics? Not as far as I know. But if you look at this picture of a coronavirus particle, remember that the genome is RNA. And the genome encodes a number of different proteins. The one of relevance here is the spike protein, here in red. So the vaccine is just that. It's that piece of RNA that encodes spike protein. So what happens is, what the drug companies do is package this little piece of RNA in an oil droplet, and then that's what gets injected into your muscle. So the oil droplet then will fuse with cells, muscle cells, and release this little piece of RNA inside. And then you're back to protein synthesis, where you have mRNA, you have tRNA, and ribosomal RNA, all collaborating to make this protein, and the protein is spike, the spike protein, because that's what the mRNA encodes. Well, once spike protein is produced, it will appear on the surface of this cell. And notice that there's a nucleus here, but the nucleus is not at all involved in this process. It all happens in the cytoplasm. So once you have this vaccinated cell that's producing spike on its surface, Now the immune system comes into play. The B cells and the T cells react to this antigen on the surface of the vaccinated cell and make antibodies against this spike protein. So these are secreted antibodies. So when the vaccinated person is exposed to a virus itself, the antibodies are right there that are specific for spike protein on the surface, and they bind. They sit there and block the surface. Now, if we go back to the beginning, how the virus actually attacks a cell, you remember the spike protein has a receptor in the membrane of a human. The receptor is called ACE2. And the spike protein binds to that, and that causes an invagination of the membrane, and the virus enters. Then the cell is infected. But now we've got antibodies that are binding to the surface. So this interaction is blocked. If there's antibodies sitting on the spike protein, it can't bind to the receptor, and there's no infection. So that's how mRNA vaccines work. Is that clear to everyone? And I should emphasize that mRNA vaccines are new. As far as I know, this is the first time mRNA has been deployed as a vaccine, and it's very curious that 2 pharmaceutical companies have adopted this for coronavirus. So then several people raised the question, well, if mRNA works so well, why do we have an adenovirus vector? Why is that necessary? Well, I don't know about motivations of pharmaceutical companies, but my guess is that different companies have their preferred technologies. I know this to be true. So some companies prefer a viral vector, and since mRNA is a new technology, it's risky. So Pfizer and Moderna decided at the outset to risk trying to develop mRNA as a vaccine. And they've done so. The upside is that they work and they're very efficacious. The downside is that because they have to package the RNA in these little nano droplets of oil, they're very fragile. And because of that fragility, they have to be stored in ultra cold conditions, -80 centigrade. And it requires 2 doses to achieve full immunity. You get some immunity with 1 dose, but you really need 2 to be fully protected. Now, if you contrast that with another company, Johnson & Johnson, which apparently is more comfortable with the viral vector, which is what they used. They used adenovirus, reverse transcribed spike into DNA, and spliced that into the virus, and that is the vaccine. The advantage of the J&J vaccine is that it's much more stable. It can be stored in a normal refrigerator at 4 degrees Fahrenheit. And it only requires 1 dose, and that's a big advantage. Somebody asked about this third one. We can talk about that a little later, but first, let's contrast what we've just said about mRNA with how a vectored vaccine works. So again, you have the virus particle, you have the mRNA genome, you have this little piece that encodes spike, and you want to get this into a cell. So how do you do it? Well, adenoviruses are very good vectors. They're very good at getting genetic information into host cells. And so you can't you can't propagate RNA in a DNA virus. So what do? You reverse transcribe, and I've got that illustrated here. You reverse transcribe the mRNA for spike into double-stranded DNA. And that DNA is now compatible with the adenovirus. So now the virus has a double-stranded DNA encoding spike. When that is injected into muscle, you have an endocytosis going on. The virus binds to the membrane. It invaginates, takes the virus in. Now, nothing happens in the cytoplasm immediately because this is DNA. So what the virus is capable of doing is injecting its DNA genome into the nucleus of the host cell. And there, the DNA is transcribed to RNA, which then leaves the nucleus. And go to the next picture. Now we're back to mRNA. So it's exactly the same from here on. The mRNA is translated into spike protein. Spike protein goes to the surface of the cells, inspires antibodies that block any incoming virus. So the thing to remember here is that there is this enzyme called reverse transcriptase, which can be used in the lab to convert RNA to DNA, and that makes it compatible with a DNA viral vector. So are there any questions about the differences between the two types of vaccine, how they're produced, what the principle is? Okay, we're out of time, so I'm going to have to hurry along here, see what else might be important to say. Someone asked, why get the vaccine if you're still going to be susceptible to the virus? This is not true. If you get the vaccine, you're protected. How does the vaccine protect other people that you contact? Well, once you're vaccinated, the virus cannot propagate and you cannot copy itself. And therefore, you don't have a viral load and you can't disseminate because you don't have virus to disseminate. So the people that you contact are not going to get inoculated by virus from you. Someone asked if you're symptomless, do you still need the vaccine? Yes. From what I've read, even if you've contracted the virus and you're symptomless, it's still advisable to get the vaccine. Why are taste and smell often lost in patients? Well, it's because the olfactory receptors in your nose are unusually sensitive to this virus, and so their function is impacted by the disease. Will warm weather stop the virus? Well, it's pretty much the same phenomenon as with any flu type virus. Warm weather tends to reduce the transmission for a number of different reasons. And some people ask about vectors. Can mosquitoes or ticks vector coronavirus? Not to my knowledge. Does anyone know? Has anyone heard about vector other than transmission through droplets from people's mouths and noses?
Not as far as I'm aware.
Yeah, well, okay. Me too. Will COVID completely disappear worldwide? Probably not within a reasonable time frame. There will always be a certain amount of propagation. If you just think about smallpox, there have been attempts to eradicate it worldwide for a long time. and it's essentially eradicated, but it still pops up now and then. So it's really hard to get a virus to completely disappear. Can a virus, this is an interesting question, can a virus mutate to non-virulence? Think about that. How would you know? It would be propagating in your body, in somebody's body, causing no symptoms. So if a virus doesn't cause any symptoms, it would be hard to recognize it as a new mutation. How to detect a mutation? Sequence the DNA or RNA. Okay, I've gone over time. Time will tell whether this exercise has been useful for you. Any questions about this?
Could we get this uploaded to Canvas?
Yeah, make a note to do that. Yeah, that's a good point. It may be useful for you when you study to think about some of the questions that you yourselves have asked and what the answers are. Anything else?
I just want to verify that the exam has been moved to next Wednesday, correct?
Next Wednesday and next Monday is a quiz. A quiz on the last 3 topics we've discussed. The test 2 will cover everything since test 1.
Professor,
Yeah,
this is Kit. Will test number 5 be part of the second exam?
say that again.
Will test number 5 be part or be included of the second exam?
Number 5.
Yeah, test number 5.
Oh, will quiz number 5 be included? Yes.